Consider an animal smaller than a grain of salt, with 8 legs and a mouth like a needle. We call it the tardigrade, the water bear, and under normal conditions it lives an unremarkable life in a film of water on a patch of moss. But in 2007, European scientists dried a batch of these creatures into dormancy, strapped them to the outside of an unmanned satellite called Photon M3, and exposed them to open space in low Earth orbit. They met the raw vacuum, the cosmic radiation, and the full, unfiltered glare of the sun's ultraviolet light. When the capsule came home, a portion was still alive. Against the vacuum alone, survival was nearly complete. Against cosmic radiation, still high. The solar ultraviolet was the great killer. Only a small fraction endured its harshest dose. And yet some of those that came through went on to lay eggs that hatched. This was not a machine. It was a living thing whose metabolism had fallen to nothing and then resumed. The trick begins with drying out. As the tardigrade Ramazzottius varieornatus loses its water, it does something strange. It floods its own cells with proteins called CAHS until they crowd the cytoplasm. As the water vanishes, the leading idea is that these proteins self-assemble, and the cell's interior sets into something glass-like, a stiff, vitrified scaffold. Researchers stress that the exact physical state, true glass or gel or network, is still being worked out. The proposed purpose is to immobilize and physically shield the fragile machinery within, to hold delicate molecules still so that being still, they cannot break. The animal is not asleep. Its metabolism drops to undetectable levels. It has become, in effect, a tiny pane of biological glass, waiting. We can watch the glass form and dissolve in a laboratory. Purified CAHS protein dissolved in water is a fluid. Dry it or concentrate it, and it stiffens, reversibly, into a vitrified gel, a fibrous network, exactly as the model predicts. Researchers have taken these proteins and expressed them in bacteria, in yeast, and in human cells, and those cells too became more tolerant of drying out, partially, not perfectly. And the space experiment, led by Swedish and German scientists under the European Space Agency, confirmed the headline consequence. A dormant organism, properly prepared, can briefly endure conditions almost nowhere found on the surface of the Earth. The implications may reach further than one peculiar animal. If life can be paused and preserved in glass, then researchers hope that blood products, vaccines, perhaps one day transplant organs, might be stored dry without the fragile cold chain medicine now depends upon. That remains a goal, not a technology, proven in fragments, not yet in full. And there is the older, stranger thought. Scientists note that a creature surviving brief space exposure is consistent with one requirement of panspermia, the speculative idea that life could, in principle, survive transfer between worlds. It is a hypothesis, not a demonstration. Real interplanetary passage involves far more than these experiments reproduce. The water bear knows none of this. It simply dries and waits, and when the rain returns, it walks away.